Well, good afternoon, everyone, and thank you for joining us today for our 5th EBT Symposium. Our talks today are about the fascinating challenge of standardizing toxicity assays. We have 3 very interesting talks for you, I hope. For those of you who don't know EBTC too well just yet, we're an organisation which is a collaborative network whose mission is to promote high-quality, high-impact research in toxicology and environmental health, working on raising research standards, making sense of evidence, improving access to research, and advocating for evidence-based decision-making. Some examples of what we do include working on systematic methods for the development of adverse outcome pathways, working with editors to improve publishing standards around systematic reviews, evaluating non-animal test methods for drug-induced liver injury, for example. And then we have some upcoming papers on Bradford Hill's concept of biological plausibility and a survey of the scientific quality of environmental health systematic reviews, which we're looking forward to telling you about on future symposia. We're funded primarily by the Centre for Alternatives to Animal Testing, and we're grateful for in-kind contributions to projects from organisations such as the US FDA, Tox Strategies, Norwegian Institute of Public Health, and Scion. If you want to join the collaboration, we operate a Cochrane-like model, running collaborative projects through thematic working groups. We'll be launching a new network structure in 2022, and if you want to find out about that and how to join, I suggest you email Katya Tsayin, our Executive Director, or subscribe to our newsletter by following this link. I'll drop these into the chat in a second. So today's presentations are about the role of research and data standards in improving the validity, reusability, and findability of toxicological research, and fit very nicely into our themes of raising research standards and improving access to research. Our speakers are Sebastian Hoffman from EBTC on what we've learned about the benefits of consistency in research methods from systematically reviewing zebrafish studies. We have Kristin Ryan from NIHS on her work to improve the consistency and translatability of the findings of zebrafish assays. And we have Anne Thessen from University of Colorado Anschutz on data standards and ontologies as new tools that make it easy to find, compare, and combine the findings of multiple studies. So I hope you enjoy their talks. We'll have 3 20-minute presentations. I'll have a 20-minute panel session discussing your questions. If you do have questions, please do post them in the chat or the Q&A. The webinar is being recorded and it will be posted on our YouTube channel. So that, I say enjoy. And I hand you over to Sebastian Hoffman, our first speaker.
Thanks, Paul. Let me share my screen. Do you see my slides?
Yes, I do.
In presentation mode as well?
Indeed, yes.
Yeah, okay. Yeah, welcome everybody. Let me kick off this last webinar of 2021 of the EBTC with the work that kept us busy for quite some time, and that is a systematic review to compare chemical hazard prediction of the zebrafish embryo toxicity test with mammalian prenatal developmental toxicity. So you may ask yourself why we embarked on such a project. We felt that it would be timely to explore if systematic review can be applied to the comparison of toxicological test methods motivated by the clinical field, what's done there in a diagnostic test method assessment. We had test method assessment expertise in-house and domain expertise available to us on various toxicological endpoints and test methods. We wanted to have a topic where there were substantial amount of literature available to make sure that we end up with some evidence to process through all the steps of a systematic review. And of note is that in toxicology, direct comparison studies that run 2 tests in parallel are hardly available, if at all. So that's a caveat. We had 2 sets of objectives. The first 2 bullet points here are our methodological objectives. So we wanted to demonstrate the feasibility of applying systematic review to test method comparison. And along the way, we wanted to identify and hopefully also solve methodological challenges. In addition, we didn't want only to work methodological, but also wanted to provide a comprehensive summary for a field where this was lacking. And considering our motivation and objectives, we ended up in choosing the Zebrafish embryo toxicity test for prenatal developmental toxicity as our test, as one of our tests, and to compare that against the standard prenatal developmental toxicity tests. Very briefly, a bit of toxicological background. Prenatal developmental toxicity can be defined as environmental insults interfering with the homeostasis, normal growth, differentiation, development, and behavior of the fetus. Of course, we're primarily interested in the human fetus, and traditionally, potential prenatal developmental toxicants have been identified through animal experiments, particularly for chemicals. And there are standardized guidelines, for example, from the OECD, available since 40 years, I guess. Yeah. And with regard to chemicals, some of you will be aware that some regulations around the world require even 2 species to be tested. The most common for chemicals are the rats and rabbits. And the main reason for that are interspecies differences between animals, and definitely also between animals and humans. On the other side, there's the Zebrafish embryo toxicity test, which has been developed about 20 years ago. At least the first works were conducted at that time. And here's a very brief and experimental setup of the Z. You expose zebrafish embryos in the first 6 hours post-fertilization, and then until 5 days post-fertilization, 120 hours, you observe various outcomes in the zebrafish related to mortality or malformations. You do that, as I said, over time, and you test usually a several concentration. Here you see just an example that on the lower right, the zebrafish is that has been exposed to the highest concentration, and the latest time point observed shows clear malformations. Not going into any great detail of the pros and cons of the zebrafish embryo toxicity test, but the pros listed here are mainly around practical issues. So the translucency of the zebrafish allows ready observation of outcomes. Its oviparous development is an advantage, and it's amenable to higher throughput systems. The cons listed here, which are discussed, is the metabolic differences compared to other species, the control of the exposure. So when I expose in the aqueous phase, how much of my substance gets really to the embryo, and there's a lot of experimental heterogeneity around. With regard to the methodology, we wanted to apply standard systematic review methodology as much as possible, but we were from the very beginning aware that that needed to be adapted here and there along the way. And here on the left-hand side, it's a fine-grained graphic of the steps of a systematic review. So once we had our planning in place, we started to frame the systematic review question, and that went through several iterations. But in the end, we decided on what's on this slide here. So what is the performance of the zebrafish embryo toxicity test in predicting the presence and absence of malformation variation in guideline studies of prenatal developmental toxicity in rats and rabbits? So you see, the zebrafish is our index test, the mammalian tests are the comparators. We were very generic and inclusive on the outcomes. We just talked about malformation and variations. And if you wish, the population were the chemicals to be assessed, to be addressed in the review. Then we defined our comprehensive literature search. And here, if you're familiar with systematic review, here's one of the major adaptations compared to standard methodology, as we are looking at 2 test methods, and there are no studies that test the 2 in one study in parallel. We had to look at the zebrafish literature first, and then go into the mammalian in a kind of second or 2-tiered review process. Not going into any great detail, but starting with the zebrafish study, we had about 10,000 studies, which after title and abstract and full text screening boiled down to 342 studies that were included. And in those, we identified almost 1,500 chemicals that were tested. This amount of chemicals was not feasible to be taken forward into the mammalian wing of our review. So we selected 75 chemicals for which we hoped that there was that it would cover the chemical space very well, but that there would also be data available from the mammalian studies. And here we then conducted a second literature review. Again, screened title and abstract and full text, and ended up with only 37 mammalian studies included. And in these studies, there were only 24 chemicals covered, 24 of the 75. That needed, of course, to go back to the zebrafish side, and we looked for those included zebrafish study that tested at least one of these chemicals, and we identified 32 zebrafish studies that had on these chemicals. With regard to the selection of the evidence, so doing the titled abstract screening and the full text screening, basically on the mammalian side, we defined that studies had to meet current regulatory standards, to keep it short. But on the zebrafish side, we had to be a little bit broader, because known from the literature, and you see 2 references here on the right-hand side, we could anticipate that there would be heterogeneity between studies, mainly with regard to the exposure, to the outcomes assessed, and the data analysis. Therefore, we defined rather broadly applicable criteria to screen or to select the evidence on the zebrafish. One of the most important one was that we categorized outcomes into 3 categories of mortality, general and specific embryo toxicity. We didn't specify any eligibility criteria with regard to data analysis, but we defined in our protocol that we will apply a very simplified data analysis to identify positives or negatives within the zebrafish studies. Here's a little bit more detail on our results with regard to the reasons for exclusion at the full text level. And I would like to draw your attention to the red rectangle here. You see that at the full text stage, about 1/3 of all the studies were excluded because they didn't meet our basic experimental requirements. So they either tested less than 3 concentration, or exposure was started after 6 hours post-fertilization, which we had defined as exclusion criteria. Taking you now to the data extraction. First, the study characteristics. On the top, you see the zebrafish, and this table is just the first 5 lines of a rather lengthy table, but it serves to show the heterogeneity between the studies. Again, in the red rectangle, you see that exposure, onset, and termination differed considerably between studies. Also, the chorionation status, in some studies, the chorion was removed, in others not. And again, in others, it wasn't reported at all. And as well, you see that the time points of outcome measurement differed between the studies. So here you see the first proof of the heterogeneity we anticipated in the zebrafish studies. And the same happened on the outcome level. Again, this is just a snippet of a rather long table on top. And you see that in the red rectangle, there are 3 studies on acetaminophen, and they differed quite considerably in their results. You see here the 3 buckets of outcome categories: mortality, general, and specific embryo toxicity. And you see fundamental differences between the studies. In the second one, you had already effects at rather low concentrations, while in the first study, you needed to go, you needed to use concentration 100-fold higher to reach these, to see some effects. And then at the last one, you see there were no effects, but we in the end decided that was inconclusive because they didn't test high enough concentrations in that experiment. You also see here the effect of our simplified data analysis. Although there were really fundamental differences between the first 2 studies, in the end, we judged them both as positive. This is just a very brief excursus. We also conducted a study appraisal, and the red and the yellow, which you see predominantly here on this slide on both the zebrafish and the mammalian literature, indicates that reporting was very poor and that we couldn't assess, due to the poor reporting, many of the risk of bias criteria we wanted to assess. And this prompted us in the end to conclude that both bodies of evidence were at high risk of bias. Here's a slide summarizing our data analysis. On the left-hand side, you see the table with the 24 chemicals which were included and on which we had data. You see then the zebrafish studies, all of them rated as positive or negative. We had repeat studies, for example, for all-trans retinoic acid. So this required us also to summarize the data across those studies, which was relatively simple for all-trans retinoic acid. That gave an overall plus, but it was a bit more difficult for other ones. And on the right-hand side of this table, you see the corresponding mammalian study results for rats and for rabbits. From this, we then distilled 2x2 contingencies tables. And you see here, to compare the zebrafish with the rat, we only had 17 chemicals, which was one of the major limitations of our review, the small sample size we had to deal with in the end. However, we concluded that the zebrafish embryo test has some potential to identify mammalian prenatal developmental toxicity, but also this zebrafish appears to be highly sensitive, as you can see here. Of the 6 chemicals negative in rat, 5 were identified as positive in the zebrafish embryo test. This brings me to conclusions and to my conclusions, and the conclusions are focused on the topic of today. If you want to see the full set of conclusions, please go to the open access publication here on the upper right. So the conclusions were that we really faced a lot of challenges that were related to the selection of the evidence. And as I've shown you, the zebrafish embryo toxicity test studies often failed even the minimum requirements we had defined in the eligibility criteria. What we also complicated our review was at the different level of standardization of the test methods. While the mammalian studies are regulatorily standardized and accepted for many years, that the situation for the zebrafish embryo test is different. So, and that leads to the fact that the zebrafish seem to be tuned very sensitive. So they want to pick up all the bad guys and rather err on the conservative side. In addition, the zebrafish embryo toxicity test appeared very sensitive due to our simplified data analysis. So there are two things coming together here. Another area of challenges was the data extraction and the data analysis. And as I mentioned a couple of times, there's a lack of that standardization with regard to time points, exposure outcomes. But you will hear more about that in the next talk. And something I didn't talk about a lot or at all, but we also looked at the zebrafish studies at an individual study level, and there were tremendous differences related to the certainty associated with those studies. And what made the whole exercise really difficult was the poor reporting quality, not only of the zebrafish, but also of the mammalian studies. And interestingly, there wasn't any time effect present in those studies we looked at. So there was no correlation between the year of publication and the reporting quality. And let me zoom out a bit again here at the end with my last slide on the recommendations. We concluded and recommended that a fundamental change in the reporting of research is needed, not only in the zebrafish, but probably in general in toxicology. That change can only be brought about if we increase awareness of the importance of reporting completeness. So authors, but also reviewers, publishers, maybe even funding bodies need to be aware of the fact that research output can be put at maximum use if it's reported in a more complete way. And with that, we also recommended that approaches to improve the ability of computers to read and process the research literature would be really needed in order to enable not only systematic review, but as I said, maximize the value of the research being conducted. And this is my last slide, and I don't want to pick out anybody in specific to acknowledge, but in the right-hand side, you see we had a lot of volunteers, and they were really essential in order to get all the heavy lifting done over the last year and finalize the project. Thank you.
Excellent. Thank you very much, Sebastian. Our next speaker is Kristin Ryan from NIHS. So Sebastian, stop sharing screen, and then Kristin, you should be able to share now.
All right, can you hear me okay?
I can hear you fine.
All right, and I will share my screen, and
that seems to be working.
Slideshow.
And you look good to go. Okay, good. Right, you've got 20 minutes. Thank you.
All right, thank you. So good morning or afternoon, depending on where you're at. I guess I just wanted to start off by thanking Sebastian and Paul for inviting me to speak and organizing the webinar. As Paul mentioned, the topic of my talk today involves our organization's approach to harmonization of toxicology testing in zebrafish. Briefly, I just need to disclose that the information in my presentation today does not represent any policies or determination for my employer. But let's get started on zebrafish and a little bit of background. I know we've already heard a little bit about this, but zebrafish are already being used widely for toxicology testing as well as drug discovery research. And I think a lot of that's because zebrafish are a fully sequenced vertebrate model with high homology to several species. They share about 70% of genes with humans, and 84% of genes known to be associated with human disease do have a zebrafish counterpart. As a vertebrate, zebrafish have many of the same organs and tissues as humans. And because of their smaller size and shorter life cycle and fairly straightforward husbandry, one can evaluate the zebrafish's entire life cycle as a whole organism or focus on certain life stages. And the utility of zebrafish for toxicity testing is certainly expanding across the globe, with hopes that this model species can serve as either a chemical screening tool, be used to predict adverse health outcomes, or even replace rodent models used for risk assessment. And although several studies have been published on assessing the teratogenicity of chemicals using zebrafish, such as Sebastian just showed you all, there remains considerable variation in the testing methods, lack of knowledge regarding chemical distribution, and inconsistent informatics approaches, which unfortunately can lead to discrepancies among zebrafish tests using even the same exact chemicals. And this reduces the power of integrative analysis and could reduce the confidence for their use in applications like risk assessment. This is problematic for organizations such as us at the NTP, who don't actually conduct zebrafish research, but are hoping to use this model for safety or toxicity assessments. We want to apply it and use it and have confidence in there. And I think a lot of other organizations feel the same way. So how did the NTP get involved in this type of research? So we are an organization that was established to do a lot of things, actually. We test chemicals of public health concern. We try to develop or validate new and better integrated test methods, provide needed information to regulatory or research agencies, and strengthen the science base in toxicology. So we felt it was appropriate for us to better understand if this model would be a good fit for our own testing program. And so we established a research program called Seize It in 2015, which included some NTP staff and local collaborators, including Nigel Walker, John Hamm, and Tamara et al. Were some of the originators of this. The goals of the program are really to determine if the model was ready to be applied to large-scale toxicology testing and identify how and when it could be used appropriately. And in doing so, we hope that our knowledge be shared with the broader toxicology community. And so the Seize It program evolved after an initial information gathering session, which included investigators in the academic industries, government, and a variety of other industry labs that routinely use zebrafish for chemical toxicity testing. And we learned a lot through our discussions with them, through workshops and literature reviews, and published a summary of some of the major sources of variability across zebrafish protocols for developmental toxicity specifically. And knowing that it might be almost impossible for each lab to walk away from that and use identical experimental designs and protocols, we focused our attention on two parameters which were deemed to likely influence chemical potency in this assay. One was the presence of the chorion. The chorion is that outermost membrane which overlays the embryo during the first 3 days of development. It's a highly fenestrated aspect of the zebrafish development with pores, so it can affect chemical access to the embryo based on molecule size, physiochemical properties, or ionic charge. Other parameter that we were interested in was related to chemical dosing. So some study designs expose chemicals one time early in development, while others do media changes every day, giving chemical renewal. So you can see for probably both of these parameters, their alterations could certainly influence chemical potency and be a source of inconsistent results across labs. So to test these theories, we designed an interlaboratory study, which I'll explain in the next few slides. Additionally, we learned that there was considerable variation in nomenclature for endpoints and data collection and analysis procedures. So we deliberately incorporated parallel research to focus on these aspects of harmonization. And all this is really to gain knowledge, share our experience, and disseminate best practices information, not only for ourselves, but the zebrafish and tox community at large. So our goal here, just to reiterate, was to assess key experimental variables proposed to influence chemical-induced developmental toxicity. And we hypothesized that Chorion removal, which is a protective barrier, and repeat exposures will increase the toxicity of chemicals regardless of other assay protocol differences that we maybe can't control. So our approach, we identified 3 labs to participate in a contract work environment. We reviewed their lab-specific protocols and cataloged their similarities and differences. We provided requirements for study conduct in 2 phases, and also very important, which I won't talk about much today, is providing requirements for data reporting. We found this to be wildly different in our previous and early experiences with data collection. So phase 1 was a basic range finding study with 40 chemicals. We provided these ourselves to the labs in a blinded fashion. And phase 2 was providing those same 40 chemicals again with an adjusted dose range to do an intra and interlab evaluation across 4 experimental scenarios. And we also requested these labs to provide all their raw data, normalized data, and the metadata, as well as their own study interpretations, so we can learn from our analysis, which is sort of unbiased because we don't have, a traditional plan for these types of analyses compared to what these labs traditionally do. And so this is just a schematic view of the interlaboratory study. 4 to 6 hours post-fertilization, labs either decoordinated the embryos prior to chemical treatment or left that chorion on. In addition, the labs either renewed the exposure media daily for 5 days or only on day 1. And after 24 hours, we did an assessment, I'll try to use my pointer here, an assessment of mortality. And then at 120 hours, we would assess the larvae for adverse phenotypes as a result of chemical exposure. And here we just have one example of a control fish that's been treated with DMSO only. And as you increase concentration of a chemical, you can see that we're able to identify concentration dependent increase in various malformations. And then other aspects of the studies, which we kept similar across the labs, or really tried to include things like tests being run in triplicate on different days, including positive and negative controls, and also the set of vehicle control fish. So presently, phase 1 is complete. We've identified several opportunities to apply standardization or harmonization to this data. We established a data analysis pipeline. We're applying standard ontology terms and creating a web-based tool for visualizing the data in a variety of ways. Because some of these aspects of zebrafish harmonization are often overlooked, I'm only just going to spend a few minutes on each of these activities within our program, but I'd be very happy to get an email from anyone listening asking more about this, because each of these are very large projects. And while I'd love to share final phase 2 data, these studies are still in progress, but I think within the year, a lot more is going to happen and we will be able to get some data out to the public very soon. And so to begin, I'm going to focus on our data analysis approach and pipeline, which was led by my colleague, Ray Washi. So after receiving the raw data from each lab, the incidences of developmental effects are summarized as 2 different types of toxicity endpoints. We have percent mortality, or the percent of dead embryos, and the percent affected, which is the percent of embryos either dead or positive for any adverse phenotype or malformation. The response of the percent affected endpoint is always greater than or equal to that of the percent mortality. And in this example, you can see that this chemical did not induce mortality at the concentrations tested, but it did cause malformations as observed by an increase in the percent of affected embryos at a lower concentration range. And so next we conducted Bmc modeling to identify the concentration at which the chemical caused mortality or malformations for each chemical tested in each lab. So the approach to calculate a Bmc versus a different metric of toxicity is really based on our experiences modeling dose response data from in vivo and in vitro assays, and the acceptance of this model by risk assessment groups and regulators. Just a few more quick notes about Bmc modeling. The Bmc, I'm probably preaching to the choir here, this group probably already knows this, but Is a lot like obtaining a lowest effect level or a low L. However, it does utilize all of the concentrations tested as well as the data variability to pinpoint the concentration at which you start seeing a chemical specific effect. And to estimate that BMC, you first need to understand the variability in your data and assign that as a baseline response level. We call that the benchmark response. For this project, we used the data from just the DMSO treated fish to understand the level of variability in basic mortality and malformations in those fish. And once we identified that value, we calculated the concentration above that line, which I have here in red, to cause a significant increase, and that's assigned as the BMC. We also looked at the confidence around these BMCs, focusing more on the BMCL or the lower bound limit. So here's just a snapshot of how some of the data looks with 4 chemicals that were tested in one laboratory. One of the first things you can see is that the data are fairly consistent within a lab and within a chemical across 3 runs designated by this pinkish color, the blue and the green. However, I wanted to be very clear that this wasn't always the case. We had a lot of data variability with certain chemicals and not others. And what was really nice about this project is that we were able to have telecons with the labs to discuss the nature of the variability and try to identify if it's assay specific, chemical specific. Really nice collaborative work looking at just even the basic data from the range finding study. In this plot, I have just a little bit more data. I want to highlight some points here from 3 labs that are designated as lab A, B, and C with different colors. And that horizontal line you're seeing here at the top represents the concentration range tested by each lab. The overlaying black dot represents that BMC, which is an average of 3 runs. And just a reminder that BMC is the concentration at which the zebrafish are affected in the form of either mortality and or malformations. So in some instances, we see that the chemical was negative across all labs, no need to calculate a BMC. In other cases, we snuck in some duplicate chemicals, which were blinded, the labs didn't know, but we were very pleased to see that for these duplicate chemicals, we saw fairly consistent BMCs being recorded within and across labs. That was really nice to see. There were some cases where you have an active chemical only in one lab, or perhaps a case where you have a chemical active in all 3 labs, but that BMC shifts. And so you can ask questions, why might that be? Is it something special about their protocol? But diving deeper into that wasn't averaging all the data. What about comparing individual chemical-induced changes in phenotype within and across labs? And is there even any consistency? So this was something we hadn't really embarked on yet. We wanted to go through that since we had collected all this data. And so to do that, I'm just going to highlight my point very quickly with this previous example. So for each of these cases, knowing which chemicals caused which kinds of malformations or spectrum of malformations could really be valuable for learning more about the mechanism of action of that chemical, or perhaps incorporating that data into various weight of evidence approaches for risk assessment. So for example, what if the BMC for chemical 6 was not only different for each lab, but perhaps the most sensitive DevTox endpoint for lab A was based on malformations in the eye and snout, whereas you had a curved or bent tail in lab B and you had delayed hatching in lab C. And for chemical 4, where lab C maybe identified heart edema after exposure, but labs A and B didn't see anything. So you might immediately think that there's a problem with the assay, but what if a lab doesn't even screen for those same endpoints? This really wouldn't be a fair comparison across the labs. And so we had to do some work to really assess the data that we had and whether or not they were comparable. And so for assistance with this, we turned to Anne Thessen, who is our next speaker, and Melissa Heindel to learn more about how ontologies can help us harmonize the terminologies of these malformation or phenotype endpoints. And in fact, our discussions with them, including the scenarios I just mentioned, helped us design a secondary project regarding inconsistencies with zebrafish screening that Anne's going to talk about next. And so we had the opportunity to learn a lot from Anne and Melissa. I, for one, wasn't even aware of the power of ontologies and how useful they are for sharing information using a common language or conducting queries for data comparisons. So in this simple example over here on the right, it's often the case that 4 different labs might see or might use the same chemical, but unfortunately categorize observations with 4 different terms. And this could be pretty challenging for somebody like myself, who's not a zebrafish expert, or perhaps a regulator who isn't used to seeing this, and to know that these are all the same findings. Essentially having these harmonized ontologies is very helpful for this approach. And I just wanted to point out that this is quite lacking currently in the community. So this is just a quick table, how we identified in our dose range finding that each lab had different terms, lab-specific terms, and we worked with our ontologists to identify ontology labels for each of these using the Ontology lookup service website. So we could harmonize terms and find out where we had consistencies and where we didn't. Here's an example of looking at craniofacial malformations across the labs. And again, if you weren't particularly looking for this, you might miss the fact that all the labs are looking for something similar, but you might just not know that. And so I won't get into this today, but the application and use of standard ontology terms can unfortunately reduce the granularity of some of the information, and this needs to really be discussed among the scientific community. So the very last part of my talk is going to be about our development of a web-based tool. This is just in the early stages, so I'm just going to mention that we're developing this free tool because we want people to have all the information from our study, including why we tested these chemicals, experimental differences and similarities, learn about general ranges of variations within assays and compare results across labs, and hopefully eventually collate all this data to compare it to rodent studies and human evidence for further translation and risk assessment. We do have some experience doing this. In 2019, we released a tool called DNT Diver. And this was really to focus on neurotoxicity endpoints with 10 different labs. Within this, we also had some zebrafish data already, so we kind of understand how we want to present the data and show it. And just as by way of example from our diver that we currently have, we can compare relative potencies of flame retardants run in 26 different assays. And we view this as a starting point to ask questions, think about prioritizing chemicals. So last slide, I just want to summarize that, our group here at the NTP aims to support the broader adoption of zebrafish models in toxicology. We want to do this by identifying which experimental parameters have an influence on tox outcomes, but also reiterate the importance and the value with harmonizing all aspects of these studies, including collection, analysis, and reporting. And because I am at time, I'm just going to say there are so many people to thank. This is a highly collaborative project among many staff at NIEHS. We have great contract support. And then really, we wouldn't do this without the actual zebrafish researchers that have been involved. And this is a list, and it's probably even grown since then. But thank you for your time. And I can take any questions at the end. Thank you.
Fantastic. Thank you, Kristin. So I'm going to just hand us over now to our final presenter, Anne Thessen, who will be talking about ontologies and zebrafish assays. So Anne, I think you should be able to share your screen. And you are on, yep, there we go. So we can see it. Can we hear you?
You should, can you?
Yes, we can. Yes, we can. Now.
Okay, and I have not been allowed to start my camera, so you're just going to look at my slides. Okay, hello everyone. Good morning, good afternoon. I'm going to speak to you today about developing semantic technology for high-throughput zebrafish studies. I'd like to start by thanking the organizers for inviting me to speak today. And I also like to start with my acknowledgements. First, I'd like to thank all of my collaborators in the Translational and Integrative Sciences Laboratory and all of my collaborators in the CZIT project, which Kristin mentioned in her talk, particularly the BioBide Lab, the Tengue Lab, and the Line Lab. And of course, all the survey participants who participated in the results that I'm about to show you. And this work was funded by NIH and NIEHS. So when I talk about semantic technology, what I'm really talking about is an ontology. An ontology is a formal computational representation of knowledge in a particular domain. It's made up of things or entities, classes, and the relationships between those classes. Here in this diagram, we see a picture of a small portion of the cell ontology. And we see that each of these classes are represented by the gray box. They each have a human readable label and a persistent, unique, machine-readable identifier. The relationships in this ontology are ISA. And so we can see here that an efferent neuron is a neural cell, etc. But these relationships are much more than just an arrow and a phrase, ISA. These relationships are also accompanied by a series of formal logic rules that a special piece of software called a reasoner can compute over and use. And so while a human can know that an efferent neuron is a type of neuron, if you model that knowledge in this way, then a computer can also make use of that knowledge as well. And so an ontology or semantic technology allows for logical inference and sophisticated data queries. And if you'd like to know more about what ontologies are, how we use them, how we build them, please see this talk here at this bit.ly link. And so a knowledge graph, which I'm sure you've heard much more about knowledge graphs and ontologies, a knowledge graph is just another way to represent the same type of data. It requires slightly different modeling techniques. While ontologies are community-driven schema that move very slowly, knowledge graphs can grow much more quickly and be much more responsive to user need. And there are far more tools and services available for working with knowledge graphs than with ontologies. The knowledge graph that I'm going to focus on today is the Monarch knowledge graph, and you can interact with this graph at monarchinitiative.org. This graphic shows the contents of the Monarch knowledge graph. You can see that it is built of several different data sources that contain lots of different types of data. And each of these data sources is mapped to an ontology or a standard. And then we mapped those ontologies or standard to what we call unifying ontologies like Eupheno and Mondo. And it's these ontologies that we brought together to make the actual knowledge graph that you can interact with at the URL. And so all of this basically combines, it integrates genotype, phenotype data across species. And so we did this for some specific use cases, particularly rare disease diagnostics. Now, not all diseases manifest themselves in the same way in everyone. And when you're dealing with a common disease, a physician can see several different patients and get a feel for how the disease presents itself and what's a common symptom and what's a rare symptom. But if you have a rare disease, the physician does not have all of those data points. And so what happens is that people with rare diseases can often wait for years to get a diagnosis. But in the Monarch knowledge graph, we have disease profiles that link a specific disease, in this case, Wiedemann-Steiner syndrome, with its suite of symptoms or phenotypes. So this is the disease profile for this particular syndrome. Here we have a patient, a 14-year-old boy, and another patient, a 3-year-old girl, and this is a list of their symptoms or phenotypes. You can see that each of these patients has a profile that only partially overlaps with the disease profile and only partially overlaps with each other. But we were able to help them get their diagnosis of Wiedemann-Steiner syndrome by using the graph and doing something called fuzzy matching. And this is where we can take groups of phenotypes, like a patient profile, and compare it to a disease profile using something called semantic similarity analysis. And this is made possible by the structure of the knowledge graph and the structure of the ontologies that underlie the knowledge graph. For example, so the system can know that a long toe and a short toe are both subclasses or both types of toe abnormalities. Okay, so that's like, if you remember the cell ontology, it would be long toe is a abnormal toe, or that microcephaly and macrocephaly are both abnormal head phenotypes. And so what we get then is, instead of this is the same, you know, it's instead of saying, okay, this is the same or this is not the same, you get like a probability score to say that this is the most similar thing. And this really helps with rare disease diagnostics, and we've been successful at helping people in this way using the graph. And so I've talked a lot about diseases and genes and phenotypes, but one thing that is not very well represented in the graph is environmental exposures. And right now we get our exposure data from only one source, and it's not really being used to its full potential. So that makes our graph much less useful for diagnosing diseases that have an important environmental component. And so the first step in order to make use of these data is to try to come up with a good model for representing the data and then bringing that data into the rest of the graph. And so we first wanted to start by taking a closer look at the data that we need. And one of these types of data is high-throughput zebrafish studies. And I don't have to go into too much detail with this crowd here, but high-throughput zebrafish studies is where you expose an embryo to a chemical, and then you just look at the endpoints. And this is done, you do this with a lot of embryos, and you have to use a microscope and look and make assessments very quickly about what you're seeing. Different labs often use their own methodologies and their own vocabularies to record these data. So that makes integrating data across laboratories very difficult, if not impossible. And so that's a challenge that we would have to overcome. Also, here's a model showing the proposed integration with the rest of the Monarch knowledge graph. So it's, for example, this box shows the data that you would get from a high-throughput zebrafish study. Let's say you have exposed a zebrafish embryo to aldocarb, and then you observe the microcephaly endpoint. Well, you can use zebrafish and microcephaly as hooks to link that data from your study to the rest of the graph and get the context, additional information about genes and processes and diseases that might be related to your data. So that's fairly interesting, but as I said, we have a data integration problem. And that's because different labs use different terms for the same endpoint and report endpoints with different degrees of granularity. Now, ontologies are a potential solution for this because, as I pointed out before, the different levels of granularity of a phenotype are all logically linked to each other within the structure of the ontology. And so the more granular terms are related to the more general terms. So that's a way that we can handle that problem. And at its very basic level, ontologies can be used as a controlled vocabulary of sorts. So can we standardize endpoint reporting in a useful way with ontologies? And so to do this, we came up with an experiment to have participants take 2 surveys, wherein we showed them pictures of zebrafish and asked them to annotate. And in the first survey, we just give them a blank space to do their annotations. And in the second survey, we provide them with ontology terms that they can pick from to score the embryos. So our hypothesis was that using the controlled vocabulary, which is the ontology, will increase consistency in endpoint reporting across laboratories. In survey 1, we showed them pictures and asked people to score them as they would in their lab. And then survey 2, we asked them to score them as they would in their lab, but use terms provided from the zebrafish phenotype ontology. And so this is what survey 1 looked like. It was just a Google form with an image and then a blank area for them to do the scoring. And we had 18 participants, and we did get feedback regarding uncertainty about darkness of the image and the age of the embryo, which probably had an impact on the kinds of endpoints that people were reporting. So we got a lot of free text answers. And this is an example of all of the strings of annotations that we got related to an abnormal jaw. And so these terms were translated into the most similar, the most granular applicable ontology term. And those were the terms that were provided in survey 2. Now, when I show you the data, you're going to see that it was analyzed twice. The first time just looking at the very general term, and the second time just looking at the more granular terms. So for example, we got a lot of abnormal jaw structure, altered jaw morphology, and these strings were related to abnormal jaw in the zebrafish phenotype ontology. But we did get some more specific terms like large mandible or small mandible. And these were translated to the more specific ontology terms here, but because not everyone annotated to the same degree of granularity, we had one analysis where all of these granular terms were converted to a general term and then analyzed, and then another analysis where we kept everything as granular as possible. Survey 2, we had 15 participants. It was in a Google Sheet this time. We used the same images, but presented them to the participants in a different order. And here we have the ontology terms that were provided with their definitions, and just ask people to choose the terms that applied. And 5 annotators reported that they did not see all of the terms that they needed in order to fully annotate 19 of these embryos. And that's very interesting considering that these terms came from the answers that they gave in survey 1. And I'll talk more about that later. Okay, so survey 1 results. Annotators used over 1,700 unique term strings to describe 48 zebrafish phenotypes. Abnormalities of the gut, pectoral fin, and otic vesicles were the most heterogeneously described, and abnormalities of the heart and yolk were the most consistently identified. So first, does everyone mean the same thing when they use the same term? That's a big no. For example, the term heart edema often lumped many different types of heart deformities, not just heart edema. And so this is something that I'm going to refer to as an overloaded term for the rest of the talk, and I'll go more into that later. And I think this is why survey 2, people did not see the terms that they wanted to use in survey 2, because there were so many overloaded terms. Is everyone scoring the same images with the same endpoints? That was also a big no. Annotators did not even really agree on which embryos were normal. And so this suggests that it's not just an issue of people using different names for the same endpoints. We actually are seeing the reported endpoints change when we provide a controlled vocabulary. And so in survey 2, the main results does providing an ontology or a list of ontology terms improve consistency across labs. Yes, with some caveats. Repeatability increased in the second survey, except for the granular annotations. And I'll go into more about why I think that's true in a minute, but I think it has a lot to do with overloaded terms. But the analysis looking only at general terms did improve in consistency a little bit. But as I will say, this study was preliminary. We did have a small number of annotators. So let's look at the surveys here for a second. So this is taking a closer look at these surveys. We see red, the red data is in survey 1, the blue data is in survey 2. Looking at, we see for the general annotations for abnormal jaw, these are the annotations results by embryo. The variability in endpoint reporting decreased slightly for general annotations and increased for the granular annotations. Some embryos were more difficult to annotate than others, like for example, embryo 22, he has low concordance in both surveys. So that's interesting. Here's results by annotation. Again, the survey 1 is in red and survey 2 is in blue. We can see that the general annotations were much easier to identify. Normal embryos, abnormal swim bladders, notochords, somites, eyes, heads, jaws, axes, and trunks in the second survey than it was in the first survey. For the granular terms, it was only easier to identify yolk sac edema and abnormal caudal fin morphology in the second survey. So the more granular terms, most of them did not get more consistent in the second with ontology terms. And I suspect that what's going on here is when you list those terms with their specific name and a definition, then you really start to get people thinking, like, what do I really mean by this? And I suspect that if we had timed people, they would have taken much longer to finish the second survey than the first survey, because in the first survey, they're just going through and applying these annotations, whereas in the second survey, you're giving them terms with definitions and asking them to pick, and they're thinking about, you know, what do I really mean by this? And so the question then becomes, how specific do these phenotypes need to be? Is it okay if everyone just says abnormal head, or do we need to have microcephaly in here? And I think the answer is probably we need them to be as specific as they can realistically be. If we substituted microcephaly for abnormal head morphology here, we would of course get these results, but we would also get all of the other results that apply to any sort of abnormal head morphology phenotype. So it would make things a lot more difficult. So in conclusion, providing ontology terms gives a slight improvement in concordance, and so that means variability is reduced when ontology terms are provided, but only for the general terms. And we also suspect that what we're seeing is some of annotators focusing on different things as well. So providing ontology terms has the potential to improve consistency in endpoint reporting and improve data integration. And we really need to think more about the ontology terms that were provided to the participants in survey 2 and the analytical consequences of overloaded terms. I suspect that that's what's causing the difficulties with the granular annotations. And so do we do the general terms? Do we do the granular terms? It really needs to be fit for purpose. I suspect we need to be as specific as we can be while still keeping the high and high throughput. The whole point of high throughput is that we get a lot done, so the annotators can't spend a lot of time trying to figure out which specific annotation to give to a specific larva. And so in the future work, we're thinking about putting together a zebrafish endpoint atlas that will be community driven and informed by existing workflows. We know that a lot of individual labs already have their own sort of laboratory guide for doing these, and we would just like to take those guides and try to come up with a larger guide that can be shared across laboratories and that can help us be able to integrate data across laboratories. So if you'd like to collaborate in that, please let me know.
All right, fantastic. Thank you, Anne. I'm just going to ask all the participants to turn their videos on. It should, in theory, be possible. We've got gallery, set that to gallery. Maybe I can grab this link here, will work. Excellent. All right, that seems to all be working. And I will, that's the gallery view for me as well. Good. So we've had 3 really interesting talks. Thank you to our speakers. We kind of have 3 themes that we seem to be moving across. So Sebastian was talking about lessons about completeness of the reporting of zebrafish embryo toxicity assays. Kristin was talking about the consistency of conduct of these assays, and then to some extent Anne was talking about the consistency of recording of these assays, right? What happened in them and what was found. These are 3 kind of very interrelated themes that are difficult to untangle, but will speak to each other. So I think first probably I'll ask Anne, you talk about ontology and using them, but maybe if we start with just a quick kind of 101 in how one goes about building an ontology, right?
Right. So our process for building ontologies is very community-centric. The first thing we do is we try to be very use case driven. What problem are we trying to solve, and do we need an ontology to solve it? So that's kind of the first thing, is do we really need, what is the problem, and do we really need an ontology? Because these the ontologies are data infrastructure, and so it's a bit of a commitment to build this thing and then maintain it and expect people to use it. If you're building infrastructure, you need to commit to maintaining it so that other people can use it. And if you're not going to do that, then you have to be you have to think about who is going to maintain this into the future. And so we often will have actual workshops where we sit down with experts in multiple related fields, and we just sit in a room together and kind of hack out, wow, how this ontology needs to look. Can we all agree on what that hierarchy is? And oftentimes we'll also go through use cases where we say, these are the things that we would like to be able to do that we can't do now. And we'll also come up with competency questions. And these are questions that we would like to put to a knowledge base or an ontology where we know what the answer is. And this is a test to see if we have covered everything we need to cover. And so that, and it's an iterative process. We do our work in GitHub. And and it's an open, transparent development process. Anyone can submit an issue, and it's a long-term iterative process where we just work with the community to iteratively build out the ontology step by step toward a specific use case goal.
Thank you. So, Kristin, when it comes to zebrafish assays, what, in your experience, the details that are most important to be standardized, do you think?
I think Anne kind of hit that very nicely at the end of her presentation. The use case is probably very important. I would love to see all aspects, the design, the data analysis and reporting all harmonized, but that's not feasible for certain labs or groups of people. Like I said, if some people just want to screen very quickly to see if something's positive and negative, they might not need to have such a standardized process. But I think that if you really want to give your data to a regulator or have it used in some sort of weight of evidence approach to try to understand hazard, then you probably want to be as harmonized as possible. So you can get back to what Sebastian was talking about, comparing those results to, I guess, the standard tried and true rodent assays, or at least what's available about them right now. So I do think it's fit for purpose, but I think that the community, I hope, is talking more about it, and we'll hopefully together come up with some standards.
Yeah, so I suppose maybe one of the things that might be worth just teasing apart here is that we've got, these use cases, right? So experimental standards and like informational standards like ontologies are designed to solve slightly different problems, right? So I don't know if maybe both of you want to speak to this, but obviously there's a challenge of, what phenotypes people are looking at, right? When they're doing their assays, so selecting the phenotypes that might be of interest for the developmental embryo toxicity, you know what I mean, assay. But then there's also this challenge of getting people to call the same thing the same name, right? So do either of you want to speak to that issue?
I can start, and then anyone else can jump in. I think that it's definitely a problem, and I think a lot of people know about it already. I've been going to conferences talking about this with folks already, and it's, again, some of them say, well, we have our list in our own laboratory and we've been using it and it's working really well for screening, but everyone I've ever talked to in the zebrafish community is interested in ontologies and find it an amenable process. They just haven't, there's no one that's really kind of brought it together for someone to kind of champion that concept. And I've even talked to folks that a lot of people do automated image analysis. And when you think about even in that automated list, someone had to create that automated list for what you might see in a zebrafish. And I was talking to some of them and they're like, oh, well, our list only has 10 phenotypes in it. And I'm like, well, there's definitely more than 10. So we add more and then link those terms to ontology terms. So someone like Anne or another person doing data queries could keep linking back and forth. So I find that everyone is interested to do this. It's just not, it's not happened yet. And we definitely need a champion for that, I think.
Sounds good. So Anne, do you have anything you want to add?
So a big part of data standards is whether or not anyone will use them, right? And so a portion of the infrastructure for data, supporting data reuse and data preservation, is making these structures that enable people to continue to do what they want to do, right? Because people are going to do what they want to do. So it's the problem of one person calling a phenotype a different word than another person is actually a pretty simple problem to solve. You just build into the data infrastructure that this this name equals this name. And so that's not that big of a deal. And you can also create tools and services that can transform data from one format to another. Again, because people want to do what they want to do. So that's not that much of a concern to me. It's really being able to fit these data infrastructure processes into the existing research workflow. That's how you get people to participate and to use this infrastructure.
Okay, so I think we obviously need to ask Sebastian the question fairly shortly, but I would like to follow that up with a question about how you would envisage, or how one builds these tools into research workflows, right?
Well, you have to understand the workflows. I mean, it's a lot of transdisciplinary science and, science of team science, organizational psychology kind of stuff. But you really need, like I said, we do this work with the community. And if you're not working with the community, embedded within the community, talking to people, you're gonna miss it. You're not gonna understand what their research goals are. You're not gonna understand what their constraints are. And if you can't help them get to their research goals with these standards, then they're just not going to be used. And so the specific workflow and the specific tasks that you do depend heavily on the community that you're working with.
Okay, so Sebastian, you were talking about increased awareness of the need for complete reporting of research and how this is desirable. How do you suppose that this might be implemented?
Yeah, I think that's a good question. Anne alluded to it a little bit. You need to get to the researchers and incorporate it into their workflow. But I think there's also an educational component there. I think people need to understand the real value of their research beyond their immediate goal of publishing a paper or whatever that might be, that there's a broader value to their science potentially, and that can only be harvested if they really are clear about what they have done and how they have done it. Of course, there come in all the limitations that you're facing when you're going with your papers to reviewers and to journals that have word limits and all these things, which is still pretty, sometimes appears to me pretty old school with all the technologies around. Why should there be any word limits to papers in these days, for example? So I think there's a really a broader educational move on the researcher side, but also on the publisher side, maybe on the funder side as well, required. And that won't happen within a year or two.
Yeah, Kristin.
I was just going to say, just in relationship to that, we when we were working with various zebrafish researchers, it was very obvious to us when we handed them 40 chemicals and they gave us back data. It was vastly different than what they thought should be reported to us. And so we had to actually come up with a template for them to use to report back to us. And I thought that was really helpful for us, for them, just to learn what types of information might be valuable to others that you never thought of. They just report their standard information because that's what works for them for their publications. But the interaction and actually talking to them was really valuable. And we've gone on to use that template and share that template with many, many people. And not saying that template is the template, but I could envision something like that really helping with methodology. It was all for the methods and endpoints as well. And so I could envision something like those templates being really helpful for a community actually trying to report out all the information instead of just limited pieces here and there.
So would you say that researchers might be receptive to this then? Because I'm an edit for a journal, and helping researchers conform to complex, like conduct and reporting standards is challenging. And each research project is a little bit different in its own right. So kind of finding one-size-fits-all templates can be quite challenging, can be seen as an imposition. Is there anything in the work that Kristin, you're doing, or Anne, or even Sebastian, that you could suggest that might maybe a way of revisioning reporting standards in a way that combines with the work you're doing that actually makes it easier for scientists to write up what they're doing and have their data reused?
Go ahead.
One of you should answer this question.
I can start. So I don't know of any global efforts. I just know of several groups of people trying to standardize the data reporting and the method reporting types of information. And they're small groups, you know, groups of 5 to 10 zebrafish researchers. I'm just hoping that that expands out. I don't know of any larger groups. I know of folks like Stefan Schultz. He is someone we're collaborating with, and he was just telling me he's interested in creating a database for sharing data. And I'm assuming that database, we would like to put our data all into his database at some point within the next few years. And I think that he would probably give us a template and we would adjust, but I 100% agree. It can be in a position, it takes more time, especially when you're just trying to crank through studies. So I imagine it is a little bit of a pain sometimes.
Yeah, I agree. Time will help and to converge at some point. And it's not a problem specifically for the zebrafish embryo toxicity test. I think other test methods have similar complexity, and over the years they converge to a standard, and even to a standard that could be accepted by regulators. I think what we are doing here is maybe speeding up that process a bit and spreading the message and also, yeah, showing the incentives and the advantages of harmonizing, standardizing as much as possible.
Anne, do you have anything you want to add?
Well, I was just thinking in terms of the zebrafish atlas and what process we would go through. And I honestly think that we're not yet far enough along in that process for me to really know exactly how, you know, the specific steps for going through that. But one thing that occurred to me is that I suppose once we start using machine learning algorithms to add annotations to images, that will make things a lot easier to standardize, because then it's all coming off the same. We can just have the algorithm spit out the ontology terms that it sees in the images. And that's usually when you start getting large data sets that are much more heavily standardized, is when you get a machine involved.
So I guess I'm going to ask a last question and then let everyone go. But is there a risk then perhaps of over-standardization, perhaps? Because this is one of the things that sometimes when we hear from researchers who are perhaps a bit less convinced about the merits of approaching things consistently, that becomes over-consistent, over-homogeneous, and somehow militates against, say, discovery.
That's definitely a concern, and that's one reason why these standards, you should never think of a standard as this immutable thing. The standards have to change over time, and so you need a process for maintaining and updating a standard. You need a process for version control and keeping track of all those changes over time. So yeah, that is definitely a concern, and that's why the standard is, think of the standards as living things.
Thank you. So Kristin, I think I'm going to give you the last word and then we will wrap up.
Me? All right. I actually had just one quick, I think we're not far enough along in standardization to be worried about over standardization just yet. So I like to see maybe a couple more years of effort to get to that point, but really want to involve folks like regulators who are thinking about using this data to replace potentially animal tests or, like I said, weight of evidence or anything like that, to see maybe we can just learn what the minimum information is they need to feel comfortable, so we don't have to impose too many restrictions or too many standards, and create some flexibility. I just think that if we do it without talking to the users of the data, then we might go down a path that's maybe not as profitable for everybody, because I personally want this type of model to be accepted for regulatory purposes. So I'm have the intention of trying to involve them as much as possible so we can make it useful going forward.
Fantastic, thank you. So I'd just like to finish by thanking our speakers. I hope you enjoyed their presentations. Thank you for everybody who participated and joined us. So Kristin, Sebastian, Anne, thank you. That was really interesting. I hope everyone else enjoyed it as much as I did. And I look forward to hearing more about your work in future.
Thank you.
Thank you.
Time.